Okay, so thank you to those of you who've joined us in person. This is my first in-person seminar at UCLA. And for those of you who are joining us on Zoom, we've got our hybrid on. So I'm very excited to introduce our visiting speaker, Dr. Hao Zhu from UT Southwestern. So I met Hao when I was visiting Dallas, something like 7 years ago, and I didn't know him at the time, and then since then, kind of following his career, seeing the incredible publications and discoveries from his lab. When I got asked to be part of the regenerative medicine theme for the CTSI speakers, he was one of the top on my list because I wanted to bring him here and learn about what he's been doing. So Hao is an associate professor at the Children's Research Institute, as well as professor of pediatrics and internal medicine, I believe. So Hao is originally from China, came to the United States, studied biology at Duke. He went for medical school at Harvard, while also doing an honors thesis with Len Zahn, studying blood cell development in zebrafish and epigenetic regulation. He did a residency and a fellowship in Hem-Onc, and also did a postdoc with George Daly at Harvard. Made some really exciting discoveries about Lin28, Let7 axis in development, regeneration, metabolism, cancer, really important work. And then he was recruited by Sean Morrison to join his new institute at UT Southwestern, where he's been studying liver regeneration, liver disease, liver cancer, looking at SWI/SNF, regulators of disease, and making really exciting discoveries about the kinds of cells that repopulate the liver, and doing things like in vivo CRISPR screens, really exciting work. His research has been supported by being a CPRIT scholar, Forbeck Scholar, Mark Foundation Emerging Leader Award, as well as innovation awards from Stand Up to Cancer and the Moody Foundation. So thank you, Hao, for joining us, and we're really excited to hear about your work.
Thank you so much, Andrew, for that very kind introduction. It's really great to be here. It's really fun to be here in person and seeing the amazing lineup of speakers you've had in the past. It's such a wonderful honor to participate in this seminar series. All right, let me share. So I'm really excited to tell you about some of the work that we're doing in my lab currently. Here are my conflicts. I want to just tell you about those. So the liver, I think, is a famous regenerative organ. And I think it's on the Rushmore of really cool regenerative organs or animals, along with planaria and axolotls and newts. And so I think this is an amazing organ to try to understand how it regenerates. So my lab is really interested in trying to figure out how that normally regenerates, the healthy kind of healing in terms of surgical regeneration and chemical toxin regeneration. But we also want to use this system because it's so proliferative to try to see if we can use it as a platform for gene discovery and regeneration gene discovery, not just understanding how to do it normally, but to try to make regeneration better for the liver and other tissues. Even though it regenerates extremely well, it does have its limitations. So if you keep damaging the liver with things like hepatitis viruses and monogenic diseases, metabolic disorders, alcohol or NASH, which is fatty liver disease, eventually after decades of this kind of damage, you do end up failing. So there's a state called cirrhosis, which is the liver begins to look like a football, and it feels like a football as well. It's a very problematic end-stage liver disease. It's a kind of a common endpoint for many of these chronic liver diseases. This is a super huge clinical problem. When cirrhotic patients come to the clinic, they have all kinds of problems. They're yellow because of high bilirubin. They've built up fluid in their tummies because there's a lot of portal hypertension through a hardened tissue. and they bleed because they don't have enough platelets and they don't create enough clotting factors. And furthermore, they have brain dysfunction because they're not metabolizing all the toxins that they're ingesting. And if all of that wasn't enough, and mind you, none of these things we have targeted therapeutics for. All of it is supportive care. So if that wasn't enough, they often end up getting hepatocellular carcinoma, which is primary liver cancer, the most common form of liver cancer. And it usually arises in the setting of damage. So an overarching question is, how does tissue injury over many decades lead to dysfunction and lead to cancer? And many people are interested in this problem, but it's very complicated because there's so many different things going on. There's injury, death, regeneration, inflammation, fibrosis. So it's really a storm of different events mediated by different cells and different genetic events. And so we are trying as best we can to try to dissociate things that are adaptive and maladaptive in this process, to try to functionalize different events that are occurring to understand what helps and what hurts. We're very interested in regeneration. Does regeneration help, or does it contribute to this outgrowth of cancer? And in recent years, we've become more intrigued by the genetic diversification that occurs from mutagenesis during cirrhosis, and how the different genetics of different cells might play a role in cell competition, outgrowth of adaptive or maladaptive clones. And so I'll tell you a couple of stories around this theme. So with that in mind, so I'm going to tell you two parts of the two stories about the normal and the diseased liver. So the first part is cells that contribute to regeneration and homeostasis, and also cells that contribute to cancer in chronic liver disease. And then the second part, I'll talk to you about mutations that occur in the liver and what they mean for the health of the liver. All right, when you look at a slide of the liver, it's very, you could say that it looks a little bit monotonous. They're all mostly hepatocytes. There are bile duct cells in here and blood vessels, of course, but it's fairly monotonous. They look very similar. So you might think, well, there's one cell type and pretty much does all the same things. But the reality is a little more complicated. So the fundamental unit of the liver is the hepatic lobule. And this is cartoon-wise shaped like a sort of hexagon if you cross-section it. If in 3 dimensions, it probably looks more like a submarine sandwich or a long, elongated football. But if you cross-section it, they look like hexagons. And what happens is blood flows from the gut through the portal vein into the liver and flows down the portal, flows into the portal triad and commingles with arterial blood and flows down the sinusoids, which I'm not showing you here. And then the blood flows across different zones of hepatocytes into the central vein and back into the heart. So there is a differentiation of different zones that fulfill different metabolic functions. So when you look at gene expression in the liver, it's actually quite intricate and beautiful. This is work from Ben Zou's group showing different RNA in situ patterns for zone 1 and zone 3 related metabolic enzymes. And these beautiful patterns are actually not very unique. That when you do single cell sequencing, this is work from Shalev Itzkovitz's group, who's pioneered a lot of single cell sequencing. They find that most genes in the liver are zonated and geographically specific to different parts of the lobule. Of course, this gene expression means that there's functional heterogeneity in different parts of the liver, different parts of the lobule. And we know that from a metabolic standpoint. The question is, for one question that we're interested in, is there some heterogeneity in the regenerative capacity of these different cells or in the risk for developing cancer? Do you get more cancers from zone 1 or zone 3? Who regenerates more? And the field is beginning to sort of get excited about these questions because we appreciate that there's quite a bit of heterogeneity. So we decided to tackle this problem first, which is what is the source of new hepatocytes in the liver? And I think it's been a field that has been challenging because there's not a consensus stem cell model. It's not like in the gut where you have a crypt stem cell that rapidly proliferates in the villus. It's not like that in the liver. And I'll go through some of the competing models of liver homeostasis or regeneration. This is all under normal, non-injury conditions. one of the original thoughts is that the hepatocytes around the portal tracts, or in zone 1, proliferate and stream towards the central vein and repopulate over time. Another model that was proposed more recently is that cells around the central vein, which are in zone 3, and they're Wnt active cells, might be proliferating in the other direction to repopulate. And then there was a correction in the markets, and basically then some people thought, well, there's no difference between any cells and they're all the same. So this is the distributed model where every cell was probably contributing small amounts to regeneration. So as you can see, there's massive confusion. There had been massive confusion about who was contributing. One of the key issues is that there were just a small handful of Cree models to sort of sort these issues out, and the resolution wasn't good enough to sort of figure out these issues. So Yonglong Wei, a postdoc in my lab, took on a very big effort to try to add to the Cree models. And so he made about 11 or 12 different Cree models. And this is enabled by the fact that you can CRISPR in Cree-ER now into the mouse genome much easier than you could before. So he cloned and did all this work with the help of the engineering corps, and we can now label different populations of hepatocytes in zone 1, 2, and 3. And so when you cross these mice to tomato, add tamoxifen, you can see different patterns that are reminiscent of the RNA in situ patterns that I showed you before. It's really pretty, first of all, and that's what we want to appreciate from a very childlike perspective. So we can label these populations, and he's done a sort of a comprehensive lineage tracing approach. And I'll just summarize what we found because most of this is published. So when you label the zone 1 population around the portal tracts, over time, that population seems to contract and get smaller over time, meaning the number of cells that are labeled gets smaller. And if you label the opposite population in zone 2 and 3, that population seems to do the corresponding thing, which is to expand. And so these two results really agreed. And it told us that zone 1 cells were not streaming into the liver to repopulate. We then labeled that sort of innermost ring of cells by either negative tracing or positive tracing. And we found that over time, nothing happened. And this is in agreement with other groups that had shown this too, that there was no streaming from zone 3 into zones 2 and 1. And then we did a thing where we could leave the zone 3, a bigger part of zone 3 unlabeled, and we found that actually that seems to get smaller over time. So I told you that zone 1 gets smaller and zone 3 gets smaller, so that leaves zone 2. And indeed, when we use a tracer with HAMP2, it seems that those cells do proliferate more to increase that population. Now, this is not massive regeneration. This is very slow repopulation because the liver is a slow turnover organ. So I want to emphasize that these are subtle changes, not massive proliferative events. However, to try to explain this, we did trace proliferation over a 10-day period, and what we saw was consistent. You saw more proliferation in zone 2 than in the other zones, although the other zones do see some level of proliferation. So this could help explain why there was an expansion in that population. Now, what's the mechanism? The mechanism seemed to be obvious because when we looked at cyclin D1, and many people had seen this before, cyclin D1 was expressed on the protein level, at least, in this intricate pattern that's exactly in zone 2. So it made sense that cyclin D1 was a driver. And mind you, not every cell that's cyclin D1 positive here is proliferating, only a subset are. Cyclin D1 is doing other functions other than just proliferation. And so, however, when we delete cyclin D1, the Ki67 positive proliferation is gone. So it does make sense that cyclin D1 expressed in zone 2 is a key driver. And in data that I won't show you here, but we also think that IGF-BP2 and mTOR are contributing to the sort of proliferative impulse within zone 2. Now, I think a weakness of our prior studies was that we didn't have a great zone 2 marker. We were doing a lot of negative tracing by looking at zone 1 and zone 3. And so Yonglong had made another Cree, which is really nice, IGF-BP2. It's one of the only very sharply zone 2 genes. he knocked in Cre-ER into the 3' UTR and then crossed with tomato. And you can see that the red here does label a more faithful zone 2 population. And the green here is IF for glutamine synthetase, which is a zone 3 marker. And you can see the portal areas are generally spared of labeling. So this became a useful tool. A graduate student in my lab, Yu-Shen Lin, further characterized these to sort of validate some of our findings. And indeed, when she traced the zone 2 population, she again saw this gradual increase over long time periods, indicating that it is likely that these populations are expanding into zone 1 and zone 3. And she did more work in terms of looking at regeneration. So everything I've shown you so far is a sort of homeostatic repopulation. So in terms of regeneration, the most famous assay is the partial hepatectomy, where you cut out 70% of the liver, and then you watch it grow back within 2 or 3 weeks. And so classically, what we think is that a lot of hepatocytes are proliferating here in all of the zones. And so we think that it's unlikely that any one zone has a dominant contribution, because essentially when you go through this traumatic event, everybody pitches in to regenerate. And indeed, what she found is when you look at zone 2, you see not a dramatic expansion, but it may be a slightly increased contribution from zone 2 hepatocytes into partial hepatectomy regeneration. Now, I would like to criticize partial hepatectomy because it is a great assay, but it's also one that not all of us experience. So we think of this as regeneration, but it's not the sort of regeneration that most people face. Most people take a little Tylenol, they drink a little alcohol, they do something, maybe some drugs, and then they see chronic mild injury. And one of the ways we can model this is by giving Tylenol or different chemicals. And indeed, in this scenario, when you give Tylenol, high doses, you see that there's a elimination of the zone 3 population that's labeled here in green. And you can see zone 2 kind of fills in and rescues that. Likewise, when you injure the other side of things with a chemical called DDC, you see the opposite effect. You kill off zone 1 and the zone 2 population comes in and rescues. So altogether, I think what we're showing is that the zone 2 population has a role in homeostatic regeneration, but it's also well positioned between where most of the injury is occurring, which is in the peaks of metabolic gene expression in zone 1 and zone 3. So it does make some teleologic sense that zone 2 would be this sort of reserve population that come in for the rescue. So I have to mention that several other groups, so Bin Zhou's group in Shanghai also found similar results for homeostatic repopulation. And Holger Willenbring's group had also found, even though they suggested it was distributed, they found a higher rate of proliferation in zone 2. So I think this is a sort of a group, not a collaborative, but a group effort to find a similar result here. Now, this is great, but what about in disease? What about in cancer? So 2 guys in my lab, Andrew and Jason. Andrew is an MSTP student who has graduated. Jason is sort of taking on the taking the football and running with it. And they're trying to track malignant hepatocytes in the context of these different zones. And so I think this is a really important problem because we want to know what the earliest events are after you mutate really important genes in liver cancer. What happens to those hepatocytes that carry those mutations? We usually are not able to track that. So this would be really interesting to know what happens. So we picked beta-catenin because beta-catenin is in the top 3 mutated genes in liver cancer. It's TERT promoter mutations, P53, and beta-catenin. And we know that beta-catenin mutations alone are not enough to drive cancer in the liver, or not super efficient in driving cancer. So we looked in the TCGA and found that ARID2, which is a SWI/SNF chromatin remodeling tumor suppressor gene, was the most statistically co-mutated with beta-catenin. So we paired those 2 together to study mutant clones in the liver. And the beauty of studying beta-catenin in the liver is that there's a really good downstream marker of Wnt activation called glutamine synthetase. And that's expressed in zone 3, right in that sort of 1 to 3 cell ring right around the central vein, there's a bright IF signal of GS. And when you mutate beta-catenin in other parts of the liver, other parts of the lobule, you see ectopic GS expression. So it's one of those rare situations where you can track mutant clones without having some complicated reporter system. So Andrew took our tools, the zone 1 CRE, tomato, and beta-catenin exon 3 deletion, which activates beta-catenin, and also ARID2 deletion, and tried to make mutants in zone 1 and in zone 2 and 3 using these CRE tools. And you can see that there's a beautiful labeling of zone 1 mutant clones within the GLS model, and a labeling of the zone 3 population here. Now, what you'll note is that there's suboptimal efficiency of the CRE in these different zones, but we were okay with that because we didn't want to activate beta-catenin in every cell. We just wanted a mosaic situation, which reflects what is happening in human tissues. Now, there is a difference in the efficiency, but we don't think that is playing a major role. Okay, so we then lineage traced this situation over 6 months or so, or 6 months or longer. And in the zone 1 mutants, what we notice is that the clones sort of coalesce in zone 1, but they don't go away and they don't really expand. They sort of stay the same throughout time. Now, in contradistinction to that, in zone 3, we saw a dramatic disappearance of beta-catenin mutant clones over this sort of period. And it was very clear that by the 6-month point, there were very few ectopic GS clones that were being activated. Even within the GS positive ring, we didn't see beta-catenin mutant, we didn't see evidence of beta-catenin mutations that would be seen with like beta-catenin going to the nucleus within that GS ring. So we think that there's a disappearance of the clones. Looking closer, I'm sort of showing you what I had said before. When you have these mutant clones around the portal tracts, and then over time, they may proliferate a bit to sort of maintain that population over time. However, in zone 2 and 3, you see this sort of disappearance of those clones over time. Now, we think that there's some competition between the wild type and the mutant clones, because when we look at tomato here, tomato is labeling the wild type clones within zone 3. And so over time, the red cells that are not green take over that domain of cells. So we think that there is some sort of fitness delta between the mutant and wild type clones. And ironically, or surprisingly, the mutants do worse than the wild types. Now, all of this would make you think that the cancer should arise from zone 1 and not zone 3. However, that was the opposite of what we found. So when you looked at zone 1 mutant livers over time, we found very few tumors compared to zone 3 mutant livers. So the efficiency of tumorigenesis was much higher in zone 3. And this is quantified here. It's a little bit complicated, but you can focus on the surface tumors quantified per mouse. So each dot is a mouse. And you can see that there's a much higher efficiency within zone 3. Okay, then we took the tumors that arose from zone 1 and zone 3 and did RNA sequencing to figure out if there was a retained legacy of those zones within the tumors. And it was somewhat surprising, but we couldn't discern, if you do unbiased clustering of the RNA sequencing, we couldn't discern the zone 1 from zone 3 tumors. They were all sort of clustered together. If you forced a difference, there's a little more proliferation in the zone 3 tumors than in the zone 1 tumors, but a very subtle effect. The overarching finding is that there's a retention of zone 1 gene expression within these tumors, probably because they're Wnt activated, and zone 3 is a Wnt active domain. And then zone 1 genes, I'm not showing you here, are all downregulated in these tumors. So Jason did an experiment to try to figure out what are the genes in zone 3 that might be regulating the tumor efficiency within that zone. And so we used a bunch of different data sets, single cell and CRISPR screens. Long story short, we came up with a list of 11 genes more highly expressed in zone 3 that we then perform sort of single gene deletion models of. So there's a cool tool you can use in the liver called hydrodynamic transfection. And you can take transposons containing beta-catenin and MYC, co-inject them into the liver of a wild-type mouse, and form tumors within 2 weeks. And so it's very rapid. And you can also introduce Cas9 and guides and whatnot. So Jason individually deleted all of these genes along with beta-catenin and MYC. And this is a big experiment, but I'll try to focus on the key findings here. So the measurement of the liver to body weight ratio after 1 to 3 weeks allowed us to quantify the amount of tumors that were in the liver. And essentially what we found is when you delete CDK1, you had much fewer tumors as you would expect. When you delete PTEN, you'd have more tumors. And so this is a good metric to figure out the requirements for these particular genes. And so we decided to focus on 2 genes, GSTM2 and GSTM3, which seem to, when deleted, reduce the amount of tumorigenesis. So what are these genes? So GSTM2 and 3 are glutathione S-transferase mu family genes. They're zone 3 focus genes. They're phase 2 detoxification enzymes that conjugate glutathione to xenobiotics and endogenous chemicals as well to sort of detoxify them. They can be seen to be associated with worse outcomes in cancer. And so when he did these experiments showing deleting GSTM2 or 3, he saw dramatically reduced tumor initiation in the liver. And so these tumors are these little dots of darker tissue in here, in the control livers here, compared to the knockout livers. And similarly, when you knocked out these genes in the setting of beta-catenin and MYC, you could extend their survival akin to knocking out CDK1, significantly better than controls. Now, we're still working on what the exact mechanisms are. As you can imagine, this will be very complicated because they're affecting many, many kinds of xenobiotics. And one of the possibilities is that they're antioxidant enzymes that when you delete, they will increase oxidative stress within the liver. And through that increase in oxidative stress, they may have an effect on apoptosis. So we do see increased ROS and we do see increased apoptosis. So those are potential downstream effects of deleting these genes that might have an anti-cancer function. Now, what's interesting is that there are some drugs and small molecules that seem to inhibit these glutathione transferases. And one of them, ethylchronic acid, was developed as a loop diuretic. So a problem with this drug is that it causes you to urinate quite a bit. But in liver patients, that's not a huge problem because we need them to get rid of the fluid from their bodies. In any case, we've used ethylchronic acid in our models, and what we find is that there's a sort of a mimicking of the genetic effects, reducing the amount of tumor initiation driven by beta-catenin and MYC. So I want to summarize this sort of ongoing work, which is first, we think that zone 2 hepatocytes are preferentially repopulating the normal liver. We think that mutant cells can be outcompeted by wild-type cells in the liver, especially beta-catenin mutant cells. However, it's paradoxical that more tumors come from zones 2 and 3. And I think it's because, so this is telling us that clonal expansions, like what you see in clonal hematopoiesis, are not essential for tumorigenesis in the liver. And I think there's a competitive process of trying to cull mutant clones in zone 3, but the sort of natural antioxidant mechanisms in zone 3 could be allowing for tumors to develop, even though we're trying to cull out these mutants. GSTM2 and 3, I think, are among multiple genes within zone 3 that could be regulating this process through antioxidative stress and anti-apoptotic mechanisms. All right, and so I'm going to talk about the second part, which is about somatic genetic changes in the liver. And in fact, I've already sort of talked about somatic genetic changes, but this will be more focused on not on the cell biology, but on the mutations. So I think everybody knows now that we're not entirely wild type, that we are more or less mosaics. So this is an image from the skin, eyelids of patients who've had plastic surgery on their eyelids, where you have copious amounts of dense mutant clones recurrent in a number of genes. And then this is the same thing is going on in the esophagus and probably in many other tissues. So in the disease setting, we and others have looked at the cirrhotic livers and found that there's a lot of mutations in cirrhotic livers, as you might expect. In fact, when you see cirrhotic livers, they're little balls of clones, and all of them are mutated on some level or another. And so I think Peter Campbell had a recent paper that's really important. He profiled NASH patients, did whole genome sequencing on hundreds of pieces of liver, and he and his group found that there are many genes that are recurrently and convergently mutated in different parts of the liver, indicating that it was important for that liver to mutate those genes. And those genes are not cancer genes, they're not tumor suppressor genes, but in fact, they're metabolic enzymes that regulate insulin signaling or lipogenesis. So it suggests to us that the etiology of disease is selecting for things that are specific to ameliorating the damage caused by the disease. And this is what we're seeking to sort of functionalize and understand. So I think we're really early stage in this field where everything right now is genomic and descriptive. And I think one of the things that we want to try to add is how can we functionalize the human genetic findings, which I think are really compelling. And a huge question is, can a little mutation in a little clone, we know that it benefits the clone, but can it benefit the whole animal, or can it benefit the entire tissue? And that's important because if it can, then this might be an adaptive mutation that could be applied to the entire animal for a therapeutic benefit. So that's the concept here. And all of the work that I'm going to tell you from here on out is by Zixi Wang, who's a amazing postdoc, and he's done really interesting work here. So he started by asking the question, do mutant clones, this is a question that's obvious, do mutant clones have different levels of fitness that are nutrient dependent? And so he took a gene that's well known to be important in NASH, identified in GWAS studies. So this is NBOT7. Many people have studied this. There's no mystery about the phenotype. When you delete this gene, you get worse fatty liver disease. So we also made floxed alleles of this mouse, for this mouse. And when you give Western diet, which is a high cholesterol, high fat, high sugar diet, you see worse fatty liver disease. And so Zeke confirmed this finding. But the important question is, what if you clonally manipulate this gene in a subset of cells, not in the entire liver? What would happen then? And so I think what he tried to do was to lineage trace mutant clones versus wild type clones. And so the strategy we took, which is not perfect, but I think it's good enough for what we're trying to do, took tomato reporter mice that were either wild type or MBOT7 floxed, and then gave low dose Cree to mediate the deletion of MBOT and to turn on tomato. It's not going to be one-to-one, but statistically they're going to be together. And so after doing that, giving Western diet or normal chow for a long period of time to evaluate the clones. And so let me try to take you through that. So under wild type tomato positive conditions, labeling a subset of these clones gives you about 20 to 30% positive clones under normal chow diet. This is after 4 months of diet. And then when you give Western diet, you see that there's a maintenance of that clonal population. And that kind of makes sense because the tomato positive clones are wild type, just like the tomato negative clones. So when you add the MBOT7 mutation on here, what's interesting is under chow conditions, nothing different happens. You maintain those clones because they're not fitness disadvantaged. But if you put them on the Western diet, they go away. It's very clear that they're diminished. And this tells us there's a fitness disadvantage for MBOT7 mutant clones, as you might expect, because the cells get fat they die, and then they disappear. Even though it's obvious, I think it's important for us to do this experiment to prove that the fitness changes can be read out through sequencing studies or through functional lineage tracing studies. And so this sets up a possibility that clones with different levels of fitness can be identified using sort of mosaic assays where you can deep sequence the entire liver to figure out which clones win and which clones lose. And so the idea was not to study individual genes, but to do this in a bigger way, to do a pooled sort of analysis. And so Zeke devised a method where he could give AAVs with guide, but then the AAVs would need to integrate, because if you don't integrate the AAVs, then you can't read them out through sequencing. So he put a Sleeping Beauty transposase in here, along with a CRE for convenience purposes. And then there's transposon integration cassettes in here that allow for genome integration. So because it makes the AAV into a lenti, kind of. So the simple idea is to make a mosaic liver and then to give normal diets or Western diets, and then to understand which clones maintain and which disappear. So I'll just show you that the system looks pretty good. So you can, so this is a Cas9 inducible mouse that you turn on with docs, and then you give the AAV, and then you make mosaics. When you do it for a single guide with P10, dosing the AAV upwards will eliminate more and more P10 clones. And the same is true with tomato. So you can sort of carefully titrate the AAV to figure out the dose dependency of this effect. So we needed to do something of another proof of concept screen, which is to take genes that are known to be important in NASH from GWAS studies or monogenic studies or biochemical studies, and then to figure out what happens here. So again, we do the same sort of thing. You turn on docs, you give the AAV pooled guides, then you turn off docs to stop the cutting, and then you put them on Western diets for about 6 months, and then you grind up the livers and do deep sequencing. So this is essentially the same as any kind of, CRISPR screen, except it's in an animal and not in a culture dish. So the results were, I think the results were interesting. So the genes that mediated, when you delete these genes that mediate clonal expansion, were genes that were famous for being involved in de novo lipogenesis or insulin signaling, and they made sense. So DGAT2 and GPAM are triglyceride synthesis genes. IRS1 is an insulin signaling component. PPAR gamma and SRABP are transcriptional regulators of lipogenesis. So it made sense that all of these genes, when you delete them, would allow clones to stay non-fat and survive over time. And indeed, what we found to be particularly satisfying is that 2 of the mutations that were at the top of the screening list were also the ones found in the human genetic sequencing studies. So it suggests that our mouse study is the same as the human study, or at least there's some good correspondence there. So we focused again on this GPAM gene, which is a rate-limiting enzyme for triglyceride synthesis. And so this is one of the genes in Peter's study that was found to be mutated. And again, we did the same kind of lineage tracing experiment. Under wild-type conditions, the tomato-positive clones, because we did this for a longer period of time, 6 months, the clones seemed to disappear a little bit more. Under GPAM deficient conditions, not much of a change under normal diets, but when you give fat diets, it seemed that the clones were able to expand or maintain much better than the sort of wild type clones. This is quantified here. So wild type, you saw a diminishment. In the sort of mutant clones, you saw an increase in the clonal composition. So again, I think this is the opposite result as the M-boat, but helps prove the concept that if you prevent fattiness in a clone, you can sort of lead to clonal expansion. All right, so the good news is that it seems like the system works, but we already knew about these genes. So the question is, can we learn something new about different genes? And here I'm just going to give you some of our preliminary data, not an entire story. So we partnered with Yujin Hashida's lab, who's an expert in cirrhosis genomics, and they have lots of RNA-seq data on cirrhotic livers from hepatitis C and NASH patients. And essentially, we got a list of genes from them that are transcription factors and epi factors that are dysregulated in these disease states, in the disease liver, but also correlate with histologic outcomes like fibrosis and inflammation and cirrhosis. And so they gave us two lists comprised of 472 candidate genes in these two classes. And we screened on them, and we got a bunch of genes that we had no idea whether or not they were important, but they seemed to mediate clonal expansion. So the problem now is, what do you do with all these genes? Like, how do you figure out this problem? You could spend the rest of your life figuring out 20 genes. So I think, as I was talking with Thomas about this, the people in the liver field have a real advantage in that we can make conditional knockouts much easier. So now we've done the screen, we can either use traditional methods where you generate a conditional knockout and then study the gene in the mouse, or you can just use the Cas9 deletion method where you give AAVs and then delete in the entire liver and then do this sort of classical studies in a much more rapid fashion. And so that's what we're beginning to do. So we validated the system. So what we did was give AAV sgRNA into mice expressing CRISPR, and then gave them Western diet and sugar water for 3 months, and then evaluated their parameters. So when you took our positive control genes like IRS1 and SREBP and DGAT2, all of these had reduced liver to body weight ratios, as you would expect if you had less fat in the liver. And this corresponded with reduced AST and ALT. So this suggests that the system probably works. And this allowed Zeke to then go and take all of the top genes from the different screens to sort of knock out each of these individually. And I'm really excited about this because now we can study a lot more genes on the whole tissue level and also compare them side by side so that we're not wondering, oh yeah, the mutant is better than wild type, but where does it fit in to the whole spectrum of phenotypes? And so we're at the stage where we're looking at these interesting factors that seem to be positive here. And they essentially correlate with the amount of liver damage too. So if you have reduced liver weight to body weight ratios, you probably have less fat, you have less inflammation. And so when you look at the histology, you can look at the controls here. They're very fatty. Many of the genes don't do much, but maybe there's a modest effect. And then there are a bunch of genes that do seem to have significant protective effects against NASH. And so those are the ones we're kind of focusing on. And we just have a few clues now. So because these genes were picked with a sort of with an idea that they were going to be correlated with human disease states, they already had built-in significance when you talked about histologic sort of associations. So TBX3 is a transcription factor that probably operates in the Wnt, downstream of Wnt, that is negatively associated with fibrosis and inflammation. SMID2 is a lysine methyltransferase, both on histones and non-histone proteins, that is associated with ballooning, which is an injury response, steatosis, and fibrosis. And this is in human disease. So we're working out some of the mechanisms now, but essentially it appears that it could be that SMID is operating through beta-catenin because it's known to methylate beta-catenin. And then beta-catenin could be operating through TBX3, which is a transcriptional driver of the Wnt response. So we're working up these hypotheses now. I want to leave you with the idea that you know, our tissues are performing genetic screens. So the human liver, when it's injured, is performing its own screens on its somatic mutations that are carried in there. So I think we should also do the same thing and try to figure out what the meaning of these clones are. And it's very clear that only a subset of these clones are becoming cancer. And a lot of them have adaptive or other functionality that would be interesting. So just to conclude here, I think the important things here are that we think that we've functionalized some of the genetic discoveries made by Peter Campbell and others, showing why these mutant clones expand. And we think that some of these expansions can benefit the tissue and organismal health. And finally, I think it's a really interesting source of new genes for metabolic disease. You know, we focus a lot on germline mutations for NASH or other metabolic disease, but there's another resource here where somatic mutations could be very important. All right, I think I stopped sharing. So I just want to thank the people who did the work. Yushin and Yonglong did the lineage tracing. Yonglong did all the CREs, and Yushin is working on IGFBP2 CRE. Andrew and Jason's not in the picture, but they worked on the zone 1, zone 3 cancer models. And Zeke has done all the fatty liver screening. And of course, I want to thank our funding sources who make the work possible. So I'm happy to take any questions from online or in person.
Excellent. Thank you so much. Just really exciting work. I mean, the kinds of tools you've developed to be able to address these questions is unbelievable. There's a bunch of questions here, but I wonder if I can ask the first one.
Sure.
So in the model where you have the mutant clones, I think it was the beta-catenin and the ARID2 mutant, and then you saw kind of looking at their expansion versus when tumors develop. And what's the timing between where you looked at expansion versus tumors develop? And is that enough time for additional mutations to drive that process?
Yeah, so when we look for about 6 months, we don't see many tumors at all. So it takes 6 months, and then around 6 to 8 months, you begin to see tumor clones. And certainly, I think what I envision is that in zone 3, there's this sort of decline in the mutant population until one of the clones can find another set of mutations that allows for a rapid expansion in terms of tumorigenesis. Now, the question is that expansion a normal expansion of normal cells, or is it a tumor already? So this would be akin to saying it's like a big bang, where you have all of a sudden you get tumor. It's not that you go through these normal clonal expansions, then tumor. It's boom, tumor. So at least in the model, it takes that long to sort of see these things happen.
Mm-hmm. Great. So if you're in the room with a question, Lucy can give you the microphone. There's a question here about, I think I can interpret it that, you know, what you've learned about the regeneration in the liver, can that be used to stimulate other organs? And maybe something about CMD or.
Yeah. So that's one of the things we're excited about. Liver already regenerates, so you can only, you know, we need to help it regenerate in certain circumstances, but the pathways that we discover could be applied to other organs. So, for example, we did another screen identifying a BAS epigenetics as a target for regeneration, and we think we can apply those small molecules to colitis and sort of make colitis regenerate better too. So certainly there's going to be some conservation between organs. And because the screens are difficult to do in other organs, let's start in the liver, find the hits, and then maybe try to see if they work in other tissues.
That's great. So there's a question about actually fibroblasts. So are they, do they reside in different hepatic zones, and are they the source of fibrosis in the liver?
Yeah, so the thinking about the fibrotic source is hepatic stellate cells, which are the vitamin A storage cells in the space of discs. Those cells turn into myofibroblasts after injury and activation, and those myofibroblasts produce the fibrosis. So there's sort of a mesenchymal population within the liver. I don't know if we know that they're zonated or not, but every other cell is zonated. So probably there's going to be some zonation there. Question is that zonation super important or not? It's unclear at this point.
Right. We have an in-person question.
Yeah, so that was amazing. I learned so much. So I was wondering if you showed really nicely that the partial hepatectomy, with the partial hepatectomy experiment, that zone 2 is responsible for repopulating. Well, in certain forms of damage, zone 2 is responsible for populating. What about in diet insults? does your high fat diet influence the different zones? So that's one question.
Yeah, yeah, definitely. We've been doing those experiments where we're giving a Western diet, sugar water for long time periods. We're also doing alcohol. So we're looking at different zonal. And the story is really complicated. It's not, I can't really explain it right here because we're still figuring it out. But the contribution to regeneration is different in different injuries. So it's just not like a super clean, yes, it's zone 2 expanding in all circumstances.
Okay. And then my other question is, have you looked in viral driven HCC? Do you think that do you think that you'll get different drivers driving HCC formation in that context, different cell populations?
Yeah, that's a great question. I don't know. We haven't done any viral models because they're not that easy to do. There's not like a good hepatitis C model in mouse. There's some transgenic models of hepatitis B, but we haven't focused on them because the viruses are sort of going away a little bit. It could very well be that there's different cells that arise. Could be different mutations have different cells that are important. So I think it's a good question. We just haven't gone there yet. From the genomic standpoint, it seems like the viruses or NASH really generate the same kinds of mutations. So the mutations seem to be conserved, but I'm not sure about the cell types.
So there's a question about organoids. And are zone 2 cells the source of liver organoids? Or have you looked in organoids to see if there are zone 2 like cells as well? Yeah,
that's cool. I love organoids. We don't use them ourselves, but I think people are trying to figure out, people are trying to recapitulate zonation in organoids so that it mirrors the human system. It's not clear if I don't think anyone's looked close enough to see if they're from zone 2 or zone 1. I suspect that all zones can probably contribute to organoid formation. We don't even know if humans have this sort of canonical zone 1, 2, and so it could be different between mouse and human.
Organoids are often wind driven, so you wonder if it would kind of be the zone 3 is that the one?
Yeah, exactly.
Hi, yeah, that was so good, like super fascinating.
Thank
I
you.
guess I have many questions, but I'll just keep it to one. So I was intrigued by your GST mu23. So that seems like a very specific part of the detoxification pathway, which, as you know, has many different arms and many GSTs. Why did you think specifically the mu23 were sort of came out in your screen and not the others? So is there something specific that those enzymes are detoxifying, or?
Yeah, I don't know. I think my suspicion is that it's because we only checked those, and that if we did the experiment on 100 genes, that we'd see many more zone 3 related enzymes that are detoxifying or antioxidant that would contribute to this phenotype. So I don't think they're special in that way. It's just that they had a strong effect and they were both in the same family, so we sort of focus on them. But I don't think they're unique. But I could be wrong.
Hey, Hao.
Hey.
Little bit random, but on your list of mutations, not yours, I mean, it's accumulated data in hepatocellular carcinoma, albumin is really high. Why would albumin be mutated? What contribution would that make, or does it?
Yeah, that's a really good question.
So highly, and there's some kind of.
Yeah, we've often wondered that. So in the lung cancer, surfactant is mutated.
Really?
So it's a similar thing. And we also got albumin deficient mice and tried to generate cancer in them, and there was no real phenotype. So, but that's not to say that albumin mutants don't do anything. I just think that probably what's happening is that if you can produce less albumin, there's more energy to form cancer. But that's just my.
Oh, so in a high grade carcinoma, there's less albumin expression.
So in cancer, there's dramatically reduced albumin production, probably some. But I'm wondering in the initial phases of tumorigenesis that if you have to make less, then maybe you're more more able to transform or
It's
something.
like selective pressure against expressing
Yeah.
it. So you
However,
mutate.
in the mouse, we didn't see any differences. So
And just final question, did you do any of the cancer initiation studies specifically from zone 2 promoters?
yeah, so I was thinking that people were going to ask us that because we showed that zone 2 was important. We just didn't have a really good CRE at the time to sort of do a parallel study with zone 2. And so we didn't go there. But the CYP1A2 line has a zone 2-3 coverage. So we think that it sort of comprises the zone 2-3. So we may have to do the zone 2 experiment as well.
So the IGF-BP2
Yeah,
is your best.
yeah.
zone 2, yeah.
That's right. Zone 2 is fuzzy, right? It's like between the, and then if you fast the mice, the zone 2 will sort of expand and contract. And so it's not a perfect, it's not like a cell type.
Awesome. Well, I think we all learned a whole lot. And again, really appreciate those of you who were able to come in person, and we had a bunch of people at one point on the Zoom. So thank you, Hao.
Thank you. Cool.
Excellent.
That